Now we are going to start upon the second chapter of immunology, that is cellular components. In several components, we are going to study first our innate and adaptive immunity. Before that, I have to talk to you about the barriers for infection. If any infection attacks on the body, for example, any bacteria, so first they have to cross the skin and mucous membranes. Second, after defeating this line, they have to go with innate immunity. If they cross this, then they have to fight with the ultimate immune system, that is adaptive immunity. Adaptive immunity. So in this one is sequence. So what is innate immunity, what are adaptive immunity? So the innate immunity having some components, same adaptive immunity also having components. First here going with innate immunity. Innate immunity having neutrophils, macrophages, monocytes, dendritic cells, natural killer cells, complement system, physical barriers and secreted enzymes. On other side, adaptive immunity having T cell, B cell circulating antibodies. The mechanism in that are germline encoded. Germline encoded refer to genetic information present in germ cells, like sperm and egg cell of an organism which is inherited by offspring, and they have the constant. Okay, but in adaptive immunity, there is a variation through VDJ recombination during the lymphocyte development. It's a crucial process in the adaptive immune system where developing lymphocytes like B and T cells randomly assemble different gene segments. That V is a variable, therefore diversity, and therefore joining to create unique antigen receptor genes. V means variable, therefore diversity, and therefore joining. By these components, they make a unique and multiple antibodies for different and numerous microorganisms. Now, response to pathogen. In innate immunity, there is a non-specific response, but in adaptive, it's a highly specific and defined over time. It is a little bit fast. It responds within minutes to hours, but it is a little bit slow, and it develops over long periods memory response, and it is maybe in days and weeks. And it having no memory and it having memory. And when there is a memory, the response is more faster and more robust. In innate immunity, there is no memory of the response. For example, when microorganism come and innate immunity kill it, but it have no memory and it did not remember the strategy how they overcome that microorganism. When in adaptive, they strategize all the thing, make a strategy for fighting with this. So next time if these organisms come again, then again they will fight with same strategy. Okay, that is like memory. Now we are moving to secreted proteins. The secreted proteins are lysozymes, complement, C-reactive proteins, defenses, and cytokines. And here we have an immunoglobulin with the cytokines. These are key features in the pathogen recognition. How they will recognize the pathogens. In innate immunity, they have a tool-like receptors, and they are also having pathogenic recognition receptors that recognize the pathogen and damage-associated molecular patterns that called PAMs and DAMs. By this, there is activation of NF-κB, and by this, they will release of pro-inflammatory cytokines. And we have an example of lipopolysaccharide. In lipopolysaccharide, gram-negative bacteria. Gram-negative bacteria, we have a lipopolysaccharide that is endotoxin. It binds with CD14 macrophages, and also it will trigger first they will be go with toll-like receptors 4, and then it will be trigger and initiate interleukin 1, 6, 8, and tumor necrotic factor. Understand? And same like that, we're having more like flagellin and also peptidoglycan. Glycan. It is a presence of cell wall of bacteria. They will attach with NOD receptors, and NOD stand for nucleotide binding oligomerization domain, and they will introduce themselves to the toll-like receptor and NOD receptors. And we also having mannose, that is present in bacteria and yeast, and they will interact and release by liver, that is called MBL, mannose binding lectin. And this mannose binding lectin then activate the lectin pathway of complement activation. And also we having double-stranded RNA. In human being, the double-stranded RNA is not present, so our immune system easily detect that and attack on this double-stranded RNA of viruses or bacteria and microorganisms. Also, they having non-methylated DNAs. So also in a normal human being, there's methylated DNAs. So our immune system also recognizes and attack that thing. And after these all the things, you guys know that for the gram negative, we have lipopolysaccharide. Same for the gram positive, we have lipotacharine. Is it lipotacharide? Is it? It is for gram positive. For gram positive, we having lipotacaric acid, and for gram negative, we have lipopolysaccharide. LPS. Understand? This is all about the innate immunity, how innate immunity is going to recognize the pathogens and recognize the microorganisms by PAMs and DAMs, and there are different organisms. But the thing is then, there's a LPL, LPS, there's a peptidoglycan, mannose, anything is there, but they will give us same response to every stimulus, every endotoxin, every component of microorganism, because they are non-specific. But in an adaptive immunity, they are more specific. They are having the memories and they are activated B and T cells that subsequent exposure to previously encountered antigen. They have a stronger, quicker, and immune response. Adaptive immune response decreases with age, and is aminosis, the aging of immune system. How they will do? First, innate immunity, there the macrophages, and macrophages make the phagocytosis of that microorganism that make species of that bacteria, and then they will go to antigen presenting cells, they will go to T cell, and then from T cell they will present and T cell will act on the microorganism, like that. We will read it in detail in further topics. Now we are having our next topic, that is immune privilege. Immune privileges mean some of our organs having not as much active immune system due to some reasons, and our immune system not exposed to that part of bodies. There were some organs like eyeball, the brain, placenta, and testes. Some reasons is that there are some blood barriers due to that, and also they having not exposed to immune system as much. So these cells for chemical physical mechanism limit immune response to foreign antigen to avoid damage. There will occur from inflammatory, sequence due to weight, due to immune privilege to that organs, eye, brain, placenta, or testes. They are having less risk of allograft rejection. The risk of allograft rejection as compared to other, because these organs have less exposure to immune system. we are going to talk about a little bit about antigen presentation. The antigen presentation is in the innate system. It can be activated by the free antigens, but the adaptive system requires antigen presentation. Adaptive have to require antigen presentation by the antigen presenting cells, etc. And that pathogen must be engulfed by the cells, broken down, then that pieces of proteins are transported to surfaces and that antigen presented to T cell for activation. That is happening in adaptive immune system. But in innate immunity, the free antigens also enough for the activation of innate system. The second, there is cytokines. The cytokines are cell signaling proteins. Yeah, cell signaling proteins, and they are often released by immune cells and they stimulate the inflammatory responses in various subsets. They are having different functions like chemokine. The chemokine means they will attract immune cells. They processes cause chemotaxis. They are having interleukins. The interleukin 1, 2, etc. Can tumor necrosis factor and then can cause a tumor death. We are also having transforming growth factor. The transforming growth factor also we have interferon. Why we said interferon? Because they interfere with viral replication. And most of this you guys study like CD. And CD means cluster of differentiation. Cluster of differentiation. These cluster of differentiations are several surface molecules like CD3, CD4 and CD8 etc. and found on many immune cells like T cell and B cell used to identify cell type. Some used as receptor cell binding. So we are also having innate immune system in which there is a phagocytosis, and in phagocytosis the macrophages are hallmark, and hallmarks are also for that reason you have a neutrophils there. We have in complement, nitric acid, eosinophil, mast and basophil, already you guys know. And also they recognize molecules that are foreign, they having the pathogen associating molecular patterns, and also they use the pattern recognition receptors and key receptor class in the innate immunity is toll-like receptor. The macrophages then detect cell and mast cells, and they recognize the PAMs and secrete cytokines. In innate immunity, you guys already know about all the key features for the pathogen mechanism, but we have to know about the macrophages. The macrophages are monocytes. Macrophages are guardians of innate immunity. They produced in bone marrow as monocytes. Next, monocytes. In the bone marrow, they are present as monocytes and they circulate in blood for 3 days. And when they are entering in tissue, these monocytes become macrophages. And given, according to site of macrophages, you are giving them a name like far cell that are present in liver, they have in the microglia that are present in CNS, they are osteoclasts that are present in bone. Yes. And they are having 3 key functions. The first is phagocytosis. The first is phagocytosis, second is cytokine production. Macrophages also by producing cytokine increase stimulated T cells, and they also have antigen presentation. These are three basic and important functions of macrophages. And how they will go for the phagocytosis? The macrophages engulf pathogen into phagosome, and the phagosome merge with lysosome. And these lysosomes contain deadly enzyme, and that causes death of bacteria and viruses. And they are having the reactive oxygen species like superoxides, and that produced by NADPH oxidase. If you guys remember about respiratory bursts, and they generate the hydrogen peroxide and oxygen radicals. And the active nitrogen intermediate also used for killing of bacteria. Like for example, there was a nitric oxide with superoxide that will make a combined combination and form the peroxynitrate. Also strong compound against bacteria. And we use different enzymes like proteases, nucleases, and lysosomes, and enzymes, and that hydrolyzed peptidoglycan glycan. Secondly, some pathogens block this process like phagocytosis. It is not like that our system are going to overcome all the bacteria, but some pathogens are going to block phagocytosis process, and that is mainly by the tuberculosis. The tuberculosis modify the phagosome, and they will be unable to fuse with lysosome. When the phagosome and lysosome did not fuse, then there will be not proceed the function, the process will not proceed for the destruction of bacteria, and there will not proliferation inside the macrophages and protection from antibodies. So if you guys know about the Shedick Gashi syndrome, someone these days syndrome, there is the immune deficiency syndrome, and there is a failure for lysosome to fuse with phagosome, and this disease is a recurrent bacterial infection. What it means characteristic? This disease, the lysosome is unable to fuse with phagosome. And by that process, they are not able to kill the bacteria, and there will be recurrent infection. The macrophages can exist in several states. It is interesting. The macrophages during tablets removal, but in activated form they are more active. They are more active, and the major activators are via toll-like receptor. Look at toll-like receptors, and in which they will lipopolysaccharide from bacteria, peptidoglycan, bacteria DNA that are not methylated, and also interferon gamma from T cells and natural killer cells. They also stimulate, and the macrophages, the interferon gamma is going to attract or activate the macrophages, and it is attracted by the gamma to move to migration macrophages by complement system, including C5, 8. It is so important points. That are so important points. The interferon gamma is going to stimulate macrophages, and that are attracted by C5A. C5A. Okay. we're having different surface markers and signals. For example, the key surface receptor in the macrophages we're having. The key surface markers, like if there's a LPS, lipopolysaccharide from a gram negative, it will going to attach with CD4 T. If there's a FC antibodies, then they are going to attach with FC receptors. If there is a C3b complement, they are going to attach with C3b receptor. There are some T cell molecules next CD4, they are going to attach with MSH. MSH2. And if there is CD28, they are going to attach with B7. If there is CD40 ligand, then they are going to attach with CD14 present on macrophages. That is so important. That's why you have to remember for upcoming topics that CD4 is going to attach with MSC class 2 and CD28 with B7 and CD40 ligand with CD40. And guys, the macrophages, the key cytokines are produced by macrophages are interleukin 1 and tumor necrotic factor alpha. These are key cytokines of macrophages, and other are interleukin 6, 8 and 12. The interleukin 1 and tumor necrotic factor alpha both are increased in synthesis endothelialization molecules and allow the neutrophil to enter the inflamed tissues. The interleukin 1 is endogenous pyrogen that causes a fever. And secondly, it acts on the hypothalamus. The tumor necrotic factor alpha can cause a vascular leak in the septic shock, and there is a catalytic inhibit the lipoprotein lipase in the fat tissues and reduce the utilization of fatty acids. This is causes cachexia and kills tumors in animals. Therefore, we are saying tumor necrosis factor, and then cause intravascular coagulation DIC. So, in tumor necrosis alpha, there is a high chance of DIC disseminated intercoagulopathy. Major histocompatibility complex 1 and 2. They refer to the molecule that present the antigen to T cells. There are two types, MHC class 1 and MHC class 2. The MHC class 1 collect the peptide from cytosol that are endogenous, and the MHC class 2 collect the peptide from intracellular vesicles. Cellular vesicles, and these are exogenous. And the MHC class 1 is going to collect the antigens of viral antigens, and it is related with bacterial antigens. And secondly, they interact with CD8 and also cause cytotoxic T cells. And MHC class 2 are going to interact with CD4. They also cause helper T cells. By that, you have to remember the rule of 8. The rule of the 8 is that you have to multiply MHC class 1 and 2 or with the CD4 or CD8, but the answer is equal to 8. If you make a combination of MHC1 and CD8, we will multiply both, the answer will be 8. 8 ones are 8. If we have to make the combination MHC2 and we have to multiply with CD4, then 4 multiplied with 2 will be 8. So it is the best mnemonic to memorize MHC and MHC2 resemblance with CD4 and CD8. The answer should be 8, according to rule of 8. If we talk about MHC class 1, in MHC class 1, it is cytotoxic T cell that recognize specific viral antigen. For example, it is the MHC. This is a viral antigen that is going to present to T cell, and there are receptor T cell receptors and CD8, and we have one extra receptor that is CD28. CD28 are going to interact with any receptor of antigen presenting cell that are B7, CD80 or CD86. But here that CD8 to TCR is going to interact with viral antigen. And if we talk about MHC2, in MHC2 same the thing is there, but there will be a bacterial antigen instead of viral antigen, and there will be TCR with CD4 that will interact with bacterial antigen that is present on MHC2, and that is T cell. And we say T helper cell, and then they are having a CD4T ligand that is going to interact with CD4T present on B cell. Understand? After that, they will produce a cytokine which is activated by the B cell. There will be cell class switching of immunoglobulin affinity maturation, and there will be antibody production. And you guys know there's two relations or interactions are there. One in between CD4 and MHC class 2, and CD4T ligand with CD4T. Understand? And in this, there is CD28 that is going to interact with B7 and CD18. And second, there is MHC class 1 interacting with CD8. Understand? And secondly, if you talk on more about MHC and MHC2, if you guys remember, MHC1 is 1. So all the look I have one letter like HLA-A, HLA-B, HLA-C. So MHC2 is having 2. In his name, there were 2 letters, HLA-DP, HLA-DQ and HLA-DR. The binding, the TCR and CD8 binding with MHC. One you guys already know, and MHC2 with CD4. Answer should be equal to 8. Courtney rule of 8. The structure is having one long chain and one short chain, and in that they are having 2 equal length chains, having 2 alpha and 2 beta. Understand? And if you talk about the expression, there will be all nucleated cells, antigen presenting cell and platelet except RBC, because RBC don't have nucleates. And in MHC2 there is the antigen presenting cells. Then if we talk about the function, there is the endogenous antigen. I already told you guys, the viral or cytosolic proteins, and there is a exogenous antigen that bacteria most commonly present to CD4 helper T cell. Antigen loading, there will be antigen loading MHC in the ref endoplasmic reticulum and delivery via TAP, but here the loading is invariant chain in acidified endosomes. But MSC1, there is no acidified endosome, there is rough endoplasmic reticulum. Associated proteins are beta-2 microglobin, and they are invariant chains. If you guys focus on these structures that is given below, there is change is having of one long chain that is alpha and one small chain that is beta-2 microglobin that is associated protein of MSC class 1. And thus they having chains of equal length, alpha 2 and beta 2, alpha 1 and beta 1. They are having same length chains. It's all about the major histocompatibility, but there is a HLA subtype association. Diseases some are there, they're too easy, like B27, you have to remember that mnemonic pair. It is just the game of memorization, how much remember strong memory you have. The B27 HLA subtype had association with psoriatic arthritis, ankylosing spondylitis, IBD associated arthritis, reactive and arthritis. In simple word, you can remember the B27 can be associated with seronegative antropathy. The seronegative means they are absent of RF rheumatic factor and ACPA. ACPA, anti-saturated protein antibodies. They are DR3. The DR3, the most commonly, it is like a 3. You have to remember SLE. Like E, if you make a reciprocal of E, then it looks like 3. So remember, the DR3 is associated with SLE. And DR4, there are 4 walls in one room. One room having 4 walls. So by this technique, you have to remember that the rheumatoid arthritis. The seronegative arthropathies are associated with B27 and rheumatoid arthritis are associated with DR4. And diabetic mellitus type 1 also with DR3 and sometimes with DR4. Secondly, there are some more. There is like A3. A3 is with hemochromatosis. You guys know it's a buildup of much iron in mucous membrane. And we are also having B8 that is associated with Addison disease. Addison disease, also myasthenia gravis. Also there is a Graves' disease. So you guys having one mnemonic, don't be late. Don't be late. Dr. Addison, you will send my patient to grave. Like that. Don't be late, Dr. Addison. Otherwise, you will send my patient to grave. The grave means grave disease. My patient means myasthenia gravis. Addison means Addison, and don't be late. It B8, actually B8. Just like that. And secondly, there is in Addison disease there is adrenal insufficiency in which there will be decrease in cortisol and aldosterone. There is some other diseases like multiple sclerosis, hay fever, SLE, and Goodpasture syndrome. They are associated with DR2. And also there is Hashimoto's arthritis. Hashimoto is an old doctor. They find their Dr. 5 in DR3. Like this. But this, as I explained, that HLA Subtype association is so important. So remember that's all the thing.